was a connection between superoxide dismutase, SOD, and other processes involved in disease. And it came in a very unusual, bizarre way. Before we even discovered superoxide dismutase, a couple of scientists up the coast a few hundred miles here in California, in Mountain View, were running a little company called Diagnostic Data. And they had discovered, one was a urologist in San Francisco, the other a biochemist. They had discovered a protein from bovine liver that had powerful anti-inflammatory activity. And they had isolated this protein on the basis of that anti-inflammatory activity and then started a little pharmaceutical company called Diagnostic Data Incorporated to develop, and they hoped to get approval for this as a drug to treat inflammation. And here are a couple of papers among the earliest ones that they published, actually not the earliest, but, and they called this protein orgotine. And it was used to treat inflammation in horses, actually in very expensive racehorses that have problems with inflammation in their ankles because of the trauma and the stress on those joints. And they had gone through safety tests, and they were in the process of actually getting FDA approval for this protein as an anti-inflammatory protein. When we published our description of superoxide dismutase in 1969, it has some very unique biochemical properties. They saw our publication in the Journal of Biological Chemistry and compared it to the protein they had been studying for about 5 years, orgotine, and instantly realized these were the same protein because they had the same characteristics, the same copper content, the same spectrum. And so the identity was confirmed. They contacted me, we swapped samples, and sure enough, orgotine, anti-inflammatory protein, was superoxide dismutase. And so a huge question was, how are these things connected? Because it wasn't at all clear why getting rid of this free radical that people were reluctant to even accept had something to do with a basic process and taking place in all of our bodies, a disease process, inflammation. And the answer to that came from another wonderful scientist who unfortunately passed away about 5 years ago, also here in California at the University of California, San Diego, and at Scripps in La Jolla, just up the road. Bernie Babior discovered in 1974 that activated white blood cells, these are the cells that handle inflammation in your body. They also handle getting rid of microbes in your body, keeping your body free of infection. These cells made superoxide radical. They made a lot of it. And what occurred to Bernie, and what turned out to be absolutely true, is these white blood cells make this reactive chemical in order to kill invading microorganisms. This is the phenomenon I've referred to, someone reminded me last night, of Mother Nature making a silk purse from a sow's ear. Or if life gives you lemons, make lemonade. This free radical that most of our cells were trying to avoid making had been put to good use by some very important cells in your body, the white blood cells that keep you free of infection. And so when those white blood cells encounter a microbe, a virus, a bacterium, they eat it like an amoeba eats something smaller than itself. They flow around it, engulf it, and they bombard it with this free radical. Well, the very process that keeps you free from infection also causes some problems for you because we have diseases that are autoimmune diseases. Rheumatoid arthritis is maybe the best known of those. What happens in rheumatoid arthritis? The white blood cells get their wires crossed a little bit. Their job is to figure out what's part of you and what's not part of you. If they see something that isn't you, isn't self, they kill it, because they assume it's an invading microorganism, a virus, a bacterium. Because there are lots of parts of you to keep track of, we have autoimmune diseases when these cells get confused, they make mistakes. They see the lining of the joints in your body and they think, I don't recognize that as self, I'm going to attack it and kill it. And they try to kill a specific part of your body by bombarding it with the weapon that they have, that is superoxide radical, that they use in other situations to kill microbes. Now it's directed at the lining of your joint. So you can see there's a crossover between immunity, keeping your body free of invading microbes, and inflammation, which is what happens when these wires get crossed a little bit and the artillery is directed not at the invader, but rather at self. This was an absolutely key recognition by Bernie. I immediately did a study. Bernie was considered with the immunity part of it. I wanted to show the inflammation part of it. And so I showed in this very brief study published in the journal Science that superoxide radical could degrade the synovial fluid in your joints. The synovial fluid is like grease in a joint in your automobile. It lubricates it. It allows the joints to move. It's essential for their function. When you have an inflamed joint, if you have an arthritic joint, that synovial fluid is broken down. What I showed is that the superoxide free radical made by these white blood cells could break it down. And the SOD could prevent it. It could protect that synovial fluid from being broken down. So pieces of the puzzle started to fit together. Subsequent publication, again, I'm not going to go into detail with these, but showed other functions of this interface between immunity and inflammation, with superoxide being right there in the middle of that intersection.